Okay, we're going to go ahead and get started now. And we're looking at the second exercise in Unit 1, looking at cells of and mitosis and movement of water across a membrane. Exciting topics for a Monday afternoon. So, anybody have any questions on anything? Okay, we will not be having our first lab test next week. We will start bones next week, and we'll have the lab test the week after that. So next week is the 6th, maybe 7th, whatever. I think it's the 7th of February. And we will have the lab test on Valentine's Day, on the 14th. I think that's right. So that'll be the day of the lab test, and you'll do the lab test online. You will not have to come in and take the lab test. You'll do it online, and it'll be opened up the day like you would take it in class, only it'll be online for you. So, and it won't be as painful, but this won't be as painful as you may think. But you do need to prepare for it. That's why I'm giving you an extra week to get ready. So let's go ahead and move into today's lab. And let me share the screen. And here we go. Okay. So everybody should see the slide. It says Unit 1, 2 lab, the cell cycle, and mitosis. And these are the PowerPoints that are listed in eLearn and along with the PDF. So let's go ahead and open that up a little. There we go. Okay. So cells. We've got billions of cells in our body. They're all very complex. Some of them are very, very tiny. Some are very, very long. The longest cells in our body are the neurons that make up our nerves. Some of those cells are 3 or 4 feet long. They're still a cell, but some of them run from our hip all the way down to our big toe. That's one cell. They make up a very large nerve called the sciatic nerve that comes out of our hip, goes to our toe, behind our knee. It's about the size of our thickness of our finger. It's made up of millions of individual neurons. All of our cells are specialized in things that they can do. They can absorb, they can carry oxygen, they can make proteins, they can fight infections. So these are all very complex structures with a wide variety of shapes and lifespans. Some cells will last a lifetime. Muscle cells last a lifetime. Nerve cells last a lifetime. Some only last a few hours or a few days. But a cell, regardless of what it is or what it does, a cell is the smallest unit of life that we encounter. There's nothing smaller than a cell that can be alive. And so all of our cells that are alive in our bodies, and many of them are not alive, the ones on the surface of the skin are not alive. Like when I showed you the pictures of the cheek cells last week. If we had done that exercise in class, you would have seen that those cells were dead, because it wouldn't have, unless you, because that involves taking a toothpick and scraping the inside of your cheek. And if they were alive, it would have hurt to do that. But they're not alive. The live ones are underneath, the dead ones are being pushed up. All cells, however, are going to, of all the myriad number of cells in our body with all the functions we have, they all generally have the same basic type of structure. So, and this is a sort of a general purpose diagram of a cell. The cell has 3 parts to it. Every cell is going to have, every cell except one is going to have all 3 of these parts. Cells have a nucleus. I showed you the nucleus last week on a number of cells. Muscle cells have a nucleus, nerve cells have a nucleus, cheek cells have a nucleus, epithelial cells have a nucleus. They all have all sorts of connective tissue cells have a nucleus. The only cells that don't have a nucleus in our body are red blood cells. Red blood cells have a nucleus while they are forming, and then they kick that nucleus out before they get released so they have space to carry more oxygen. That's what red cells do. Consequently, red cells don't reproduce. They don't survive for longer than about 4 months, which is pretty long for a cell. Anyway, all the other cells have a nucleus, and within the nucleus is the DNA. All cells have a membrane around them. We call it the cell membrane. This is the outer covering of the cell. It is very restrictive as to what can go into the cell and what can leave the cell. It is comparable to our skin. And then within the cell membrane and surrounding the nucleus is what we call the cytoplasm. The cytoplasm is the cell goop, the cell sap, the material that holds all the organelles in place. The organelles are these little structures here that function like organs. They're not organs, and they're not, but they do things for the cell, like the mitochondria here, this little orange thing, generates, releases energy from stored food. These green things here, they're called the Golgi apparatus, help to collect proteins that are being made and sort them out. So all proteins of the same type are being released at one time. So they all have unique structures and unique purposes here. So we have many organelles suspended in the cytoplasm. The cytoplasm probably has the consistency of hotcake syrup, if you will. Maybe a little bit thicker, but it's still a goopy, syrupy material. And you saw the cytoplasm. I showed you the slides last week of the homemade cheek cells. Well, the area that was inside the cell membrane and surrounding the nucleus was the cytoplasm of the cell. And so that's every cell is going to have cytoplasm. So the nucleus, the biggest organelle, it is our control center. It contains our DNA. Every cell in our body that has a nucleus has exactly the same DNA. It doesn't matter if it's a liver cell, a skin cell, a muscle cell, a small intestine cell, it doesn't matter. Every cell with a nucleus contains the exact same DNA. It's just that segments of that DNA that make it a liver cell are turned on, and all the other parts of the DNA that control cell structure are turned off. But the information on how to be any type of cell in the body is found there. Everything else, but it's specialized. Liver cells are liver cells, but they contain all the DNA for all cells in the body. Muscle cells are simply muscle cells, but they also contain all the DNA for every structure in the body. So we don't have specialized DNA just for muscle cells or nerve cells. So within the DNA and within the nucleus, we have our DNA, but the DNA strands are very tiny, and they're about 6 feet long. Long, skinny strands packed full of information. And what we do is we take that DNA and we combine it with a protein called histone, and the DNA wraps itself around the protein, so it looks a lot like a candy cane at Christmas, because of the way the stripe goes around the DNA function. Looks like the DNA looks like the red stripe, and the histone and protein looks like the white stripe, and it wraps itself around there. Now, the reason it does this is for stability, because the DNA strand is very thin and very fragile, and it protects itself by wrapping around this protein. And when it's wrapped together with the protein, the DNA and histone, we call it chromatin. Now, it's still all 6 feet long, but it's all protected now because it's tied into this protein. But this is the chromatin. It's only until when the cell is ready to undergo mitosis and cell division do we see the chromatin condensing into the chromosomes. The chromosomes are simply the chromatin that forms and condenses, because it's a whole lot easier. The reason it condenses is it's a whole lot easier for chromosomes to move around inside the cytoplasm if they're condensed into smaller, thicker units. Because trying to move a 6-foot-long strand of DNA and protein is very challenging because it's all tangled and twisted around here. But if you condense it into smaller structures, it's fairly easy to get it to move. Now, a cell runs on a very specific cycle. It has a very specific cycle that it follows. The 99% of the cell cycle is interphase. Interphase is when the cell is busy being a cell. So here's interphase right there. So if we look at a cell cycle, and we start out here, and we start with interphase. We go through the various stages. Interphase lasts all the way to right about there. And then for a very tiny section in here, we have mitosis. So about 99.9% of a cell's life is in interphase, doing cell things. I'm a cell. I'm going to do cell stuff. I'm going to secrete proteins. I'm going to absorb nutrients. I'm going to release, I'm going to make insulin, or I'm going to make glucagon, or I'm going to, do this. I'm going to do that. I'm going to do something. I'm going to send signals along. Most of the time, the cell is in interphase. Some cells never get out of interphase. Muscle cells and nerve cells, muscle and nerve never leave interphase. They never divide. But for the rest of the cells in our body, we do have an opportunity for them to undergo mitosis and cell division. But mitosis is the second part of the cell cycle. This is a very, very, very brief part of the cell cycle because the cell is busy doing cell things during interphase. Right at the end of interphase, which, I remember learning what we called the stages of mitosis a long time ago when I was, the first time I ever heard this, it wasn't that long ago. Stages of mitosis are interphase, prophase, metaphase, anaphase, telophase. That's how we were taught it. Only interphase isn't part of mitosis. Mitosis is the process of nuclear replication, copying the DNA. Interphase is when a cell is just being a cell. And the amount of time spent in mitosis is minuscule, extremely short compared to the rest of the time a cell is busy being a cell. Because the cell has to do cell things. But in mitosis is where we actually physically replicate the DNA. And the cell will alternate interphase, mitosis, interphase, mitosis. If a cell is going to divide, if a cell is going to undergo cell division, it has to go through mitosis first, because cell division and mitosis are actually two separate things. It's entirely possible for a cell to replicate its DNA and never divide, because anything can happen. Okay, so here's a cell in interphase. You can see the nucleus, you can see the cytoplasm, you can see the cell membrane. And that's all you can see. You can't see any of the organelles. This is what we would have seen looking at the cheek slide. This is what we saw. Excuse me. This is what we saw when we were looking at all the tissue types last week. The cell is busy doing cell stuff, as they say, carrying out its normal activities. Now, during this time frame of carrying out its normal activities, it will be growing and it will be making proteins and releasing proteins and stuff like that. But at some point here, about a third, so if oops, let me, there we go. Now, somewhere along in here, in the cell cycle, there's a stage called the S stage. This first phase here is called the G1 phase. And then the remaining portion is called the G2 phase. And this little tiny section up here is called mitosis. Now, in the G1 phase, we're growing. Now, the whole time we're doing all this, interphase is running all along here, and we're busy being a cell. When we get to the S stage is when we duplicate the DNA. And then in the G2 phase, sort of like running as a subroutine, if you will, we're getting ready for mitosis, but we're still being a cell through this whole time here. The G2 phase is making sure that the DNA got copied correctly, and then getting ready so we go into mitosis, we can accomplish it very, very quickly. Okay, now, so that's interphase. Interphase and the cell cycle. Interphase is the dominant action of the cell while it's running through the cell cycle. Mitosis and cell division. Now, these videos here are shot in real time. So this is exactly how long it takes a cell to undergo mitosis and cell division. The cell has replicated its DNA. It's replicated, it's stored in its chromosomes. Look how rapidly the whole process of cell division takes place. The stages are prophase, metaphase, anaphase, and telophase, and it's accomplished in seconds. So if a cell is alive for 10 days, the last 20 seconds or so are going to be spent in mitosis and cell division. It will already have copied its DNA at some point. The reason we copy our DNA early is because mistakes can happen. There's always a possibility of a mistake when copying the DNA, and we need time to repair that mistake. But it is definitely not a quick, it's not a slow process. It's the amount of time spent going through mitosis and actual cell division is literally less than 1% of the lifespan of a cell. Now, something else to remember is that mitosis and cell division are not the same thing. Mitosis, the classic definition of mitosis is nuclear replication, nuclear copying. On an exam, and this is an important thing to remember, a very, very important thing to remember. If you see a question on an exam that says mitosis is, or if it says mitosis is, and one of the answers is cell division, don't mark it. Mitosis is not cell division. Mitosis is nuclear replication, meaning copying the DNA in the nucleus. You mark mitosis as cell division, it'll get counted wrong. And that, I think that shows up on one of the final exam questions. So just consider that a heads up, because some people are very, very adamant about mitosis not being cell division, even though we probably learned it that way, when we were younger, when we were in school and other places. So anyway, mitosis and cell division, two separate things. Mitosis is nuclear replication. Cell division is the physical separation of the parent cell into two daughter cells. And you can see the timing here is very, very rapid. Now, some cells replicate they have a lifespan of 1 or 2 days. Some have a lifespan of months. Some have a lifespan of years before they replicate. Some don't ever replicate. As I said, muscle cells and nerve cells never replicate themselves. Okay, now. The 4 stages of mitosis are prophase, metaphase, anaphase, and telophase. Prophase, the first one. In prophase, the chromosomes, the first thing you see is the chromosomes become visible. You can see the chromosomes now are visible in right here, inside the red circle. Those lines are the chromosomes. So there's a nucleus, there's a nucleus, there's a nucleus, there's a nucleus. But this cell is undergoing mitosis. And so in prophase, the chromosomes have condensed. The chromatin has condensed to form the chromosomes. And each chromosome consists of 2 parts. They're called chromatids. And they look something like this. Oops, oh, come on. This is what a pair of chromosomes look like. These are called chromatids, and they are held together at a structure known as the centromere. And so this is what's forming in this mass down in here now. You'll notice that the nuclear membrane has disappeared. No more membrane. So the nuclear membrane has disappeared. The chromosomes have condensed. They've already been replicated early on in the S phase of the cell cycle, and we're getting ready to go ahead and pull and push these chromosomes to opposite sides of the cell. So, metaphase, the second cycle, second step in mitosis, is probably the easiest spot. Here, the chromosomes line up on the middle of the cell. We say it's the equatorial plate. That's the line in the middle. And we see spindle fibers start to form and attach to the centromeres of the chromosomes. Now, what does that mean? Well, if you look here, this is mitosis. You can see the chromosomes are all lined up in here. But if you look here very carefully on this one inside the red circle, you can start seeing the spindle fibers forming here and here. Also sort of see it in this one here. The spindle fibers are going to attach to the centromeres of the paired chromosomes. So you have here's a chromosome, here's a sister chromatid, and here's a sister chromatid. And the spindle fibers are going to start attaching themselves here and here and be able to pull them apart in opposite directions. Now, why do we line up in the middle of the cell? Well, without going into a whole bunch of detail here, we do that so we can guarantee that the same number of chromosomes go to the daughter cell on the left and to the daughter cell on the right. In human cells, we have 46 chromosomes, 23 pairs. We want to make sure we get all 46 chromosomes to the daughter cell on each side, because we've actually replicated, we've copied our DNA. So for a very short period of time here, we don't have 46 chromosomes, we have 92 chromosomes. We have to make sure equal amounts go to the same side. You don't want to have an extra chromosome on one side and be missing one on the other side. So every daughter cell gets a complete set of chromosomes. Now, anaphase, the shortest phase in mitosis, it's very rapid. This is when once the chromosomes are lined up on the metaphase plate on the middle of the cell and everybody's all accounted for, then they move to the opposite sides very, very quickly. And this is, again, is the shortest of the phases. but it's also where cytokinesis starts. Cytokinesis is the physical separation of the parent cell. See, this is one big cell right here. And if we stop the process of mitosis, we'd have one big cell with two nuclei, and it would be non-functional. But cytokinesis will start here, right around the middle of the cell. But this is the shortest and fastest of all the stages. Let me show you just how fast anaphase is. Let's go back here a few slides. Now here we go. Look at this. Now watch this one. That's anaphase right there. And anaphase slides right into the last phase so rapidly that if you blink, you miss it. Yeah. Anaphase is when the chromosomes start to move to opposite ends of the cell. Once they get to the other ends of the opposite sides of the cell, they're in telophase, and they start closing up and unwinding and forming their new nucleus in there, and the nuclear membrane forms again, and we have two daughter cells. So anaphase is a very short phase. Now let's get back to that anaphase. So you can see we're right at, this looks like it's right at the end of anaphase because the chromosomes are here and here. They're pretty much moved all the way to opposites. You can still see the spindle fibers here. Some of those spindle fibers are going to be pulling on chromosomes. Some are going to be pushing on chromosomes. Some are just going to be pushing cytoplasm so that the cell can actually physically move apart in here. And then this is telophase or telophase. In telophase, cytokinesis finishes. We form a line through here. You can see it starting to form at this point. What happens is that we have a band of protein that forms around the outside of the cell. It's the same protein that we have in muscle cells. And that protein is called a contractile protein and starts to squeeze. And it squeezes and it squeezes and it squeezes. And pretty soon the parent cell is now divided into two distinct daughter cells. It looks, while it's going through this squeezing phase, it looks very much like one of those balloon animals that you see, you know, at carnivals and stuff, where somebody dressed in a clown suit's twisting balloons around, and then you get a dog or a cat or a dinosaur or whatever, okay? Only here, as we squeeze and squeeze and squeeze, they separate. They separate into two separate cells. So we get two identical daughter cells that have the same complete makeup that the parent cell did, but they're brand new. And their cell cycle starts all over again. The chromosomes are unwound and they disappear. The nuclear membrane is reforms. We have a nucleus, and now we look like any other cell, you know, once telophase is done. And then again, cytokinesis is the process of actually physically separating the parent cell into the two distinct daughter cells. And this part of cytokinesis right here, you can see how we have this, what we call the cleavage furrow. It's a groove that forms in the cytoplasm and actually pinches off to give us the two complete daughter cells. And their cell cycle starts all over again. Yeah. Okay. Now that is the cells and cytokinesis and mitosis, two separate things. Cytokinesis is the physical separation of the parent cell to give you the two daughter cells. Mitosis is the copying of the DNA. It's what we call nuclear replication, because we have to guarantee that each daughter cell gets the same set of chromosomes that the parent cell had. And you got two daughter cells, so you have to copy the DNA. Okay, now the next thing to look at is what we call membrane transport and permeability. In other words, how do we get stuff into the cells? How does stuff leave the cells? You know, how does the cell membrane protect us? how does our skin protect us? Well, the cell membrane pretty much does the same thing for us that our skin does. Now, our cells are covered in salt water. That salt water leaks out of our blood vessels. Our blood vessels are not a perfectly sealed tube like we would imagine. They actually have lots of openings in them, and plasma is leaking out from our blood vessels all the time. The plasma is the fluid part of our blood. Blood is, it's red because of the red cells, and red cells and the white cells and the platelets all float in the plasma, but that plasma is leaking out all the time. When it leaks out, it's no longer called plasma, it's called interstitial fluid. We call it interstitial fluid because that's what this is here, interstitial fluid, because here's a blood vessel, and because of the openings in the blood vessel, the plasma leaks out and it fills up the spaces around the outside of cells. These are cells, by the way, in case you weren't certain. Those are cells. And that fluid covers the outside, floats around them like that. And that is interstitial fluid, the fluid between the cells. Now, the interstitial fluid is filled with sodium because it's salt water, and it's mostly sodium. It's got glucose in there. The glucose that has to enter, the sugars that have to enter the cells are found in there. There are proteins and fats and hormones and all sorts of chemicals in this fluid floating around the cells. Now, the cells can get their nutrients from the interstitial fluid. Oxygen diffuses, and oxygen moves from the blood into the interstitial fluid to go into the cells. Carbon dioxide leaves the cells by dissolving itself in the interstitial fluid and then moving into the blood. It is a passive process. Now, this interstitial fluid doesn't stay there forever. The interstitial fluid will be collected by a parallel circulatory system called our lymphatic system. Lymphatic system is a series of vessels similar to blood vessels, but the lymphatic system collects the interstitial fluid, runs it through our lymph nodes where our white blood cells are waiting to examine it. White blood cells are our infection fighters. The white blood cells are sitting inside the lymph nodes, like in our armpit or at our neck or in the groin, and they examine this fluid as it goes through the lymphatic vessels to the lymph nodes, and after they clean it up, they return it back to the blood as plasma again. So it's plasma becomes interstitial fluid, becomes lymphatic fluid, becomes plasma again. It's a constant cycle here. Now, the movement of nutrients and proteins and oxygen and carbon dioxide from the blood to the cells and from the cells to the blood is a completely passive process. It doesn't require any kind of energy. Cells generally will only take in what they need. They don't take in extra. They can't store a whole lot of extra. They can store some extra glucose in the liver, but most cells don't have any place to store anything extra. They make what they need. They take in what they need. Excess will get stored. If you have a lot of sugar in your plasma, the cells will take out what they want, and the rest will go to the liver and get stored there as glycogen. What we call, our version of starch. So movement across the membranes of these cells is a totally passive process. It's like rolling downhill. There's no energy involved. You get a free ride into the cells. Stuff going into the cells is a free ride. Stuff coming out of the cells is a free ride. we say it's passive, and the term is diffusion. The term there is diffusion. The key term, diffusion, right there, diffusion. Or sometimes we have facilitated diffusion. Facilitated diffusion. Diffusion is simply movement of a substance from a high concentration to a low concentration. Movement from a high concentration to a low concentration. From the high side to the low side. Totally free. Sometimes you may need help. Sometimes you may need a special passageway to get in. Glucose will go into our cells. Sugar will go into our cells passively, but it needs help. It needs a special channel to get in, but it's still free. It doesn't cost anything. Now, other substances may require the use of energy to get in. Other substances use what's known as active transport. Active transport requires energy because usually with active transport, it's like you are moving things, moving substances uphill. You're moving what we say against the concentration gradient. Okay. Now, these are the three types of transport across a membrane. On the left, you have simple diffusion. So we have over here, simple diffusion from a high concentration to a low concentration, down what we call the concentration gradient. Concentration gradient. So we go from the high side to the low side. We go passively through the membrane, like oxygen. Oxygen loves to diffuse into the cells. You know, for example, if this was oxygen, we have lots of oxygen, you know, high oxygen levels here, low oxygen here. And we diffuse into the cell. We move from the high side, the high concentration, to the low concentration. Or perhaps we're looking at carbon dioxide. Carbon dioxide is always higher inside the cell than it is outside. So CO2 always leaves the cell. That's, you know, it's all passive. Now the second one, facilitated diffusion, this could be glucose, sugar. Sugar doesn't go into the cell easily because sugars don't fit through the membrane. This membrane here is real picky. It doesn't like to let substances come across, that particularly, you know, something as big as a sugar molecule. So the sugar will have to have its own passageway. And that's what facilitated diffusion does. It creates this structure here called a carrier protein that allows the sugar to come in. And that's what insulin does. Insulin, we've all heard of insulin. Insulin, when our blood sugar goes up, our pancreas releases insulin, so that, and the insulin will tell cells to go ahead and get busy making these carrier proteins to let the glucose come in. It's still a passive process, but it's just a way to get around this barrier, because this barrier here, the cell membrane, this is the cell membrane here, is very, very selective. It doesn't like sugar. It doesn't like any kind of carbohydrate. It doesn't like any kind of protein. The only thing the cell membrane likes is fat. And only things that are dissolved in fat can go across the membrane. So we have a workaround. We have a workaround for sugars. We have a workaround for water. Water can go across because we have a special tube just for water to go across the membrane too. Bypasses this problem with the cell membrane and goes right through it. It's like a special little pipe that goes through. These are all passive processes. But suppose, oops, we have a problem. Suppose we want to bring something into the cell, but its concentration gradient is wrong. Look at this one for active transport. With active transport, we have the high concentration here, the low concentration here, but we want the substance to come into the cell. We are moving, essentially, we're like moving uphill. We're instead of, in the first two, the simple diffusion or facilitated, it's like we're rolling downhill. It's a free ride. But in this third one with active transport, it's like we're going uphill and we have to spend energy to do that. And cells don't like to spend energy, but they will. They will actually have to pump something against its concentration gradient. It will have to pump it from the low side to the high side because whatever they want inside the cell is more important than the energy they're spending to get it there. So we have active transport, and we have to use a compound known as ATP. ATP is the cell's currency. When we spend energy, this is what we use. We burn sugar to release energy. The energy is carried by those ATP molecules. So we spend ATP to do stuff. So there aren't many things that we pump across the membrane in opposite directions, but there are some that are very, very important. Now let's move on. Now, okay. So this summarizes what I just said. Membrane transport, the movement of substances across the membrane of a cell. It's either going to be passive, which doesn't require any kind of energy, or it's going to be active, which does require energy, what we call metabolic energy. And we have to use ATP. ATP is a very large molecule that can carry energy trapped in what we call a high energy bond, that we can use that energy. So now let's move on here. Now, what about when we're dealing with solutions and stuff like that? We talked about solutions. We talked about salts and solvents and solutes in lecture today. A solution, of course, is a homogeneous mixture of two or more components, usually a solvent and a solute. The solvent is the dissolving medium, which is usually water in our bodies, and the solute is what's being dissolved. The solute is sodium chloride is usually the solute, or potassium chloride or calcium carbonate or something like that. Usually it's mostly sodium chloride, mostly salt, because all of our plasma is essentially salt water. So we dissolve salts in water all the time. Now, the cells and the water in our body is found in two places. It's found in the plasma, or interstitial fluid, or lymphatic fluid, and it's found within the cells. The intracellular fluid is the water inside the cells. So if we were to look at our, there we go. Here's our cell. Here's our nucleus. This is, make sure everybody knows it's a cell. The fluid inside here is the what that means within, cellular fluid, the water inside the cell. The water outside the cell is the interstitial, the interstitial fluid. and it usually doesn't change. The percent here of salt is usually the same as the percent here on the outside. And if there's any water movement at all, it's balanced. What goes in is balanced by what comes out. So we have essentially the salt concentration inside the cells is the same as the salt concentration outside the cells. The water concentration outside the cell is the same as the water concentration inside the cell. So interstitial and intracellular, because intra means within. Cell membranes are selectively permeable. I think I've sort of beaten that into the ground, but cell membranes are very, very particular about what goes across. Things that are dissolved in water can't go across without help. Large molecules can't go across without help. Some large molecules never get across. Things that are dissolved in fat or that are fat soluble slide right through easily, as well as really tiny molecules like oxygen molecules and carbon dioxide molecules. Things like those molecules are so tiny, those little gases are so small, they slide right through. But we say a cell membrane is selectively permeable, and meaning it is very restrictive. Some things go through easily, some things need help, some things never get through. Now, the simplest of all passive processes is diffusion. With diffusion, a substance will spread from its high concentration to its low concentration. Now, whether it's in air, or whether it's in a solid, or whether it's in a liquid, it will usually move from its high concentration down to its low concentration through down its concentration gradient from high to low. What causes it to move? Well, it's caused by the random motion of air molecules in the air. If I were to open a bottle of some odor, some chemical odor agent, maybe perfume or something, or maybe an artificial flavor like banana oil or something like that. And if I were to open it up in the front of the classroom, eventually everybody would smell the odor because the air molecules moving around would carry the banana oil molecules, and everybody would smell bananas. That's from the high concentration to the low concentration until we reach equilibrium, until it's evenly distributed. The high concentration would be right in front of me where I open the bottle, and the low concentration is the far side of the classroom, and then eventually it spreads throughout. It's passive. The banana oil molecules didn't have to do anything. They were pushed around by the air molecules moving in space. The same thing applies in water. If you add a pellet of a dye or a drop of a dye to water, it will start spreading out in the water passively based on the random movement of the water molecules, because they do. They just randomly move around and bounce around, and pretty soon they have the dye distributed until we reach equilibrium. We go from a high concentration to a low concentration passively until equilibrium is reached. So we always go from the high side to the low side. You can see right here on this illustration, high concentration of a substance until it moves down from the high side to the low side passively. It is all random. It may take a while, but it will eventually reach equilibrium and be evenly distributed throughout. If I open up my little bottle of banana oil, then eventually everybody will smell banana oil, or whatever else, almond extract or pumpkin extract, or cinnamon or whatever. It's all passive because it's based on the movement of the random movement of the air molecules in the air or the water molecules in the water. No energy is needed to make this happen. Now, when we get into the cell, diffusion will still work but we have some restrictions. A molecule can go across the membrane of a cell very easily if it is very small, or if it is fat soluble, or if it has help. If it has a carrier molecule, a specialized channel or protein that will allow it to pass into the cell. Glucose, sugars are too big to get into the cell on their own. First of all, they're not fat soluble. They are large, and so first of all, they're not fat soluble, so they can't pass through the fatty layer of the membrane, and they're too large to just sneak through around the structures of the membrane. So what will happen is that sugar will hit the membrane and stop. It won't get in. So we have to have a workaround. Sugar is too big. We have a special channel for it. Oxygen doesn't need a workaround. Oxygen slides right through. So we either have simple diffusion or facilitated diffusion. Simple diffusion is passive. I've said that before. I can't say it enough. Anything that is small enough to move through the membrane, like oxygen or carbon dioxide, or it can go through special channels. And this is passive transport right here. The fat substance, in this case, something is sliding through the membrane. It is dissolvable in fat, or it is very tiny. So now osmosis is movement of water across a membrane. Osmosis, simple diffusion of water. We move water across the cell membrane through these special channels called aquaporins. It's just a tube through the membrane that the water can go through. With osmosis, essentially, water always moves from a region of high water to a region of low water across a cell membrane. So here we see a membrane right here in the middle. Here we have a high water level and a low sugar level. Over here, we have a high sugar level and a low water level. Now, if I say that we are moving from high water to low water, consider on one side of the membrane here, the water concentration, this solution here on the left-hand side is 90% water and 10% sugar. Over here, it is 60% sugar and 40% water. Now, which side of the membrane, there's the membrane right there, and it's selectively permeable, which side of the membrane has the highest water concentration? The 90% side or the 60% side? I'm sorry, 90% water or the 40% water? Well, 90% water is the high water side, high water, and the 40% side is the low water side, because 90% water is greater than 40% water. This solution over here is 60% sugar, it's 40% water. This is 10% sugar, it's 90% water. Water will go from the high water side to the low water side through the membrane. Whatever the water concentration is on one side, and when you have when you have a membrane, one of the sides of the membrane is going to have a higher water concentration than the other. Water will go from the high side to the low side. High water equals low sugar. High sugar equals low water. Still has to equal 100%. If we're 60% sugar, then we're only 40% water. What if we're 90% sugar? Let me clean this up and try it again. How about 90% water 10% water, 10% sugar, 90% sugar. Now, which side has the highest water concentration? Well, it's this side. 90% is greater than 10%. Which side has the highest sugar concentration? This side. 90% sugar versus 10% sugar. Water will always go across the membrane from the high water to the low water. High water means low solute. Because look how much, this is the solute's only 10%. But here, if we have very low water, we have high solute. But we always go from the high water to the low water. So water will always go in this direction with osmosis. It's a passive ride. It's a free ride also. Oops, wait, back. And you can see it right here. Here's our high water, low sugar, and the water will go across the membrane into the solution where you have low water every time. Even in a solid, a gel like this. This isn't osmosis, this is simple diffusion again. But even in a gel, we will see simple diffusion. Now, what this is showing is that diffusion can occur from high concentration to low concentration based on, and the rate of diffusion is faster based on particle size. The potassium permanganate is a smaller molecule than the methylene blue, and it is in a petri dish filled with nutrient agar. It's sort of like a very thick jello, okay, without any flavoring or coloring agents or anything like that. The molecules of potassium permanganate are smaller, and they are more likely to spread through the gel than the methylene blue is. The methylene blue molecules are bigger. And so they're passively moving through this gel at a slower rate than the smaller molecules are here on the right-hand side. Again, it's simple diffusion, a passive process here. Let's go past this and that. Oh, that's good. So the last thing I wanted to say then is, even though it's a solid, it will still, well, a semi-solid, it will still work. Okay, so that is everything on the cell and the cell cycle and diffusion and osmosis for lab. Let me get us out of here. Questions on anything? Okay, then I'm going to get us out, and I will see you all in lecture likely on Wednesday. Let me get us out.